Good morning, everyone, and welcome to the SAMAC Industry Services webinar. This is also the last webinar for 2025. So today we'll be showcasing two key services that support the macadamia industry. These are the Disease Diagnostic Clinic and Field Extension Services, as well as the Macadamia DNA Fingerprinting Services. Before we begin, few admin notes. Your cameras and microphones have been automatically switched off. The session will be recorded and uploaded to the SAMAC YouTube channel. And if you have any questions during the presentation, please type them into the Q&A chat and we'll address them afterwards. We're delighted to have two speakers with us today. Diagnostic Clinic at FABI, who will be sharing insights into the clinic's work, common diseases encountered, and how to sample correctly. Ms. Melissa Reynolds from the Precision Tree Breeding Platform, who will introduce the DNA fingerprinting services, explaining how it works, the progress made so far, and how it supports cultivar identification and traceability. These services are important tools for improving cultivar selection, orchard management, and overall industry advancement. Thank you so much for everyone for joining. I'll start off with a quick animation.
Healthy orchards are orchards. But pests and diseases can cause serious losses if not identified and treated early. That's where the Disease Diagnostic Clinic at the Forestry and Agricultural Biotechnology Institute at the University of Pretoria comes in, a vital industry service for South Africa's macadamia growers. The clinic helps growers identify pests and diseases affecting their orchards, giving you the right information to act quickly and effectively. Working closely with SAMAC to support industry-wide biosecurity. The clinic also helps SAMAC monitor national pest and disease trends, keeping the industry one step ahead of emerging threats. The best part? The service is free for all SAMAC members. Submit your sample information online and access easy-to-follow guides on how to collect and send them. The FABI Disease Diagnostic Clinic, protecting your orchard, supporting our industry.
Thank you. Can everybody hear me?
Loud and clear.
Yes. Okay. So thank you very much, everyone. Good morning, and I'm glad that you all could join. I really liked the video, so thank you for SAMAC for making it. It almost feels like I don't have to share any additional info, but yeah, let's get starting. So the aim of today is to really just talk a little bit about our specific services, that of course the Disease Diagnostic Clinic, but also what we do in terms of field extension. So to get going, why do we have a clinic and what do you need to understand? Doesn't want to go to the, oh, there it is, sorry. That is, of course, we're here to help you identify threats to macadamia production. I've listed a few here. We can start off by your flower blight complex, husk rot, branch dieback, root and stem canker, sting bugs, nut borer, macadamia felted cosset, ambrosia beetles, and thrips. And that is perhaps for now just to list the most relevant or the most important ones. So why do we have a clinic and why do we do these extension services? Well, first of all, the aim is for us to help you identify these pest and disease problems. For example, if we just think about flowers, understand there are three different flower blights. That's green mold, grey mold, and dry flower. And it's important for you to understand because often different chemical products will have different efficiencies towards these different causal agents. It's also important to understand that sometimes a dried flower can be as a result of a drought, or sometimes we need to think about thrip damage. And that is why sending a sample in, confirming what the pest or the disease is important. Now, next, we also are in the process of sort of mapping the presence of pests and diseases. And here we are sort of looking long-term and mapping this over time. We are almost at the end of our first year of field monitoring. So this is where field extension comes in. And what we do, we go out in the field at least 4 times a year. We do sort of a structured but random sampling. We look for the presence of pest diseases. We also make notes of the presence of beneficials. And on these farms, we have access to their climatic data, and we also have access to their control programs. And we hope to really sort of generate data over time to really understand pest and disease trends. Then, if we now move away from what we know, is also if we receive samples, it helps us to do sort of early identification. Maybe the first time a sample comes in, we're not sure, but we can see a trend with more and more growers sharing and submitting the same type of disease material, which can tell us there's something new. And in terms of this, for example, just dry flour was for the first time recognized and identified via the disease diagnostic clinic. And also in terms of this, we also identified and confirmed the presence of polyphagous shuttle borer for the first time. Then, of course, we do also Phytophthora screening. If you are a SAMAC accredited nursery, we actually do this for you, well, free of charge. Not free of charge, but it's sponsored by SAMAC. And then over time, with us really understanding and looking at all of these samples that comes in, it helps our capacity to identify pests and diseases. So who are the important people to note? First of all, Stephen Shange is our field extension officer, so he assists with field extension, and I've listed here his email contact as well as our group's WhatsApp number. So if you feel we need to come and visit, please make use of the WhatsApp number and or email him. Next, of course, in the clinic, we have Nikki. And so Nikki really helps us with all our identifications. That's both pests and diseases. Once again, I'm sharing her email as well as the group WhatsApp number. So if you are interested in sharing a sample, what should you do? And I hope this is also sort of good news for the growers, is that we now have an option where you can make use of the courier guy, and that account is actually also paid for by SAMAC. Courier guy information, like I said, sponsored by SAMAC, so no cost to you. Very important is we need the dimensions and as well as the weight. What we need on the box is your information. How to get a weighable is you're going to need to email Nikki from Fabi, and she can request and assist you with that. Always on the box, it needs to be sent to Fabi. And in terms of Fabi, Lishan is your first point of contact. She's not directly involved in the macadamia clinic, but because we have clinics that serve various industries, we have this one point person that sort of helps us with all of that and makes sure that the correct sample goes to the correct clinic. Then, of course, once you have the weighable number, you can really drop it off at your nearest courier guy kiosk. And of course, the final thing, please make note of the QR code, is although you have requested the weighable, you also need to complete the online disease submission form for us. Okay, so for me, a very frequently asked questions is sort of what to sample. So in the picture or in the slide here, I have a flower that's diseased, I've got a nut that's diseased, and I've got a leaf that is also showing some disease symptoms. And when we think of isolating that primary causal agent, we sort of need to isolate at that point where you can clearly see the disease symptom, but we also need healthy tissue. So nuts that's already been on the ground, it's really no use for us. It's already dead nuts. Whatever you isolate there will just be your sort of saprophytes that's decomposing the material. And so that's always that almost combination between healthy and diseased material. And that is sort of ideally what we really need in order to identify the primary causal agent. Then in terms of insects, please note that for us to identify an insect, we actually need an insect. If you are interested in sending us nuts, if you want us to identify and differentiate between macadamia nut borer and felted false codling moth, please spend some time, open up the nut, make sure there's a larvae inside. We have previously received nuts, and when we open it up, there's no larvae, and the only thing that we can tell you is that yes, there is damage caused by larvae of a Lepidoptera. So please remember, we need the actual insect before we can identify. The following 2 slides is just taking you through the process, so just giving you a little bit more of information of what we actually do. You'll see we received the sample. It does go to a quick surface infection step. Why we do that is, of course, in nature, we know there are fungi that's growing inside tissue. There's fungi that grows on top of leaves, on top of nuts. And we are not interested in those endophytes or epiphytes. We are interested in the actual fungus that's causing the symptoms. And that's why we quickly do the surface sterilization step. And then, of course, once again, when we sort of plate onto cultural media, it's that zone between where the lesion stops and the healthy material. We then incubate for at least a week. Oftentimes we have multiple fungi growing, so there's a few steps of us to get to this sort of single culture growing on a plate that we then can use morphology to put it into a genus level, but we end up with a DNA sequence to make sure that we can identify it to a species level. Same is true for insects. We obviously always observe under the microscope, but then we also immediately go into a DNA barcoding. If we compare the value of DNA barcoding for insects versus fungi, with insects, we are limited. There's not so many of these barcodes available on these international databases. So we are sometimes limited in only being able to tell you it's this family or that genus. But at least over time, those databases are greatly improving. Then, of course, if we are especially worried about the sample, we do sometimes send it in to the ARC for identification, but taxonomy is becoming more and more a problem, not just in South Africa, but globally, with not that many taxonomists being available. And of course, if it's a really unknown moth, we need to send it to a Lepidoptera or a moth expert, and we need to send a beetle to a Coleoptera expert. So it's not always that the ARC can assist, but when we think it is important and we are concerned, we also do share samples with them for identification. Then one last thing, in terms of identification, just as a reminder, please note that Macadamia felted cosset is a quarantine pest. You need a specific permit for transport. So please never send a potential MFC identification directly to the clinic via the courier guy. Please contact Stephen. We are going to maybe ask you to share some pictures. We are going to take you through the steps. But what we need to do is we need to come out for a visit to your farm, because at least in our facilities, we do have the permit to transport MFC. And I think this is just really important. We really need to stop the spread of MFC as much as possible. Then one last slide, I've listed it here, insects of interest. I think we all have heard that Hylomorpha halis, so the brown marmorated sting bug, is potentially in South Africa. It may or may not be the case. We haven't had any formal sort of announcement coming through from our government, but we have seen a sort of spark more interest in people submitting sting bugs. So please continue to share. But what I have here on the slide is I just want to point out something. Cunomorpha nervosa is also commonly known as the brown or the bark sting bug. It's very common in South Africa. I see it in Pretoria all the time. We know that it is present in your orchards. And that sting bug looks a lot similar to the brown marmorated stingbug. What I can tell you, if you really zoom in onto the slide, look at the stripes on the antennae. Brown marmorated stingbug is sort of white, where it's sometimes a bit creamy, those stripes on the antenna. However, where the brown marmorated stingbug is almost orangey or sometimes even more of a bright yellow. Okay, but I don't like to rely on identifications just looking at pictures. These are pictures that I downloaded from iNaturalist. So if you see a brown stingbug and you are concerned, please submit, but also stay calm because there are at least a very common species in South Africa that also looks a lot like the brown marmorated stingbug. And then maybe our last one is then the sap beetle. So yes, it's a beetle, but it causes actually damage on nuts. So it's not an ambrosia beetle. We are currently busy with surveys. I had a phone call from a grower the other day saying, oh, he found worms in his pack house. So those worms are, of course, the larvae. The larvae can be misidentified or confused with either being macadamia nut borer larvae or even FCM larvae. This is a beetle. It's different than the ambrosia beetles. It's not causing damage on your tree trunks. It is actually potentially already entering when the nuts are still in the orchards, but in your pack house, those larvae can actually eat that entire nut up to the point that there's only powder left. So we are doing surveys. We hope to have more information about it soon, but be aware, please submit. If you see larvae in the nuts and you're not sure, it is likely a moth, a Lepidoptera, but it could also be the sap beetles. And so that's basically it from me.
Okay, we're going to start off with Melissa's animation for the fingerprinting.
Macadamia growers love their orchards, but sometimes they're not 100% sure which cultivars are which. Mix-ups can happen, especially in closely related cultivars, and that's where SAMAC and the University of Pretoria's Macadamia DNA fingerprinting service comes in. With just a leaf sample, the DNA fingerprinting service confirms cultivar identity. Before planting, growers can even verify the cultivar received from nurseries, ensuring they get exactly what they ordered. The industry is building a reference set to ensure every cultivar can be accurately identified. For macadamia breeders, DNA fingerprinting is a game changer. It allows them to confirm the parentage of new breeding populations, verifying crosses, and ensuring accurate pedigrees. With reliable DNA data, breeders can make informed decisions, speeding up development. Pollen testing are also available, helping researchers confirm pollen donors and cross-pollination in experiments. From orchard to lab, DNA fingerprinting makes cultivar identity and pedigree verification effortless. SAMAC, from leaf to lab, confirm your cultivar with confidence.
Thank you, Lisa. I almost don't have to present anymore after that. It was such a cute little animation. Thanks. Okay, I'm going to share my screen and I'm going to turn my camera off while I present because I'm working with two screens at the moment, so I'm not going to be looking at you. But okay, great. Okay, so I'm going to give you a bit of an update on our Macadamia DNA fingerprinting service. So I am the manager of this platform. Sorry, I see we have people waiting in the lobby. Okay, so I'm the manager of the precision tree breeding platform at the University of Pretoria. And what we do is we specialize in translating DNA marker related research outcomes into tools that can be used by tree breeders and growers in South Africa. Our platform consists of 6 people. We've been operational for 18 years, and in that time, we have fingerprinted over 115,000 trees, and these are predominantly from the forestry industry. Okay, so the things that we are involved in is we support the research activities at the FMG EPI program at UP. So this is a program that was founded very much focused on forestry. So FMG stands for Forest Molecular Genetics Program, and EPI is the Eucalyptus and Pine Pathogen Interaction Program. So our research has very much so focused on forestry, and we have supported that. But many of the tools and the applications and approaches that we use in forestry can be translated into macadamia. And that is why we are able to add quite a bit of value, because we've established a strong base of experience and techniques at UP. Then we focus on working alongside students and researchers to take the outputs of their research and to translate these into usable tools. So that would be a DNA fingerprinting panel or a molecular breeding technique or something like that. We provide a research service to the forestry industry, as I've mentioned, and the majority of our work is actually dedicated to forestry. Probably less than 10% of our work is involved in macadamia or any other species that we work on. And in forestry, we work predominantly on eucalyptus and pine, and then we're also expanding into wattle at the moment. And in 2022, we began extending our service into the macadamia industry. And I'll tell you a little bit about the research that we've done so far and where we're at with our development. So we have quite extensive facilities at UP. In 2020, we established this high throughput DNA extraction facility. This is funded by the Technology Innovation Agency, and it allows us to extract around 800 or just under 800 samples in a morning. So it drastically increased our capacity, and we have the capacity to extract samples from about 13,000 samples per year, and we also have the opportunity to increase that. So we have a lot of capacity to expand at this point. Okay, so what are DNA markers and how can they help the macadamia industry? So the little video at the beginning covered quite a lot of that, but I'm going to delve a little deeper into this. Okay, so if we look at DNA markers, simply put, these are specific known regions in the genome where genetic variation occurs, and we can capture that variation and use it for various applications. When we look at a specific tree, the combination of genetic variants at these known positions, or the DNA markers, throughout the genome is unique to that tree. And when we combine these different variants throughout the genome, we can generate an individual's unique DNA fingerprint. So this can be used for various applications. Predominantly, we use this for identity and relatedness testing. So if we consider a specific tree, we know that its genome is unique, much like humans have unique genomes. And when we vegetatively or clonally propagate material from this tree, say in rooted cuttings or grafting onto rootstock, these would be 100% matched to that original tree. We also know, as with humans, trees have seed parents and they have pollen parents. And much like humans, they share around 50% of their DNA with each of their parents. They also share around 50% of their DNA with full sibs, so that would be trees sharing the same seed and pollen parent, and around 25% of their DNA with half sibs, so that would be sharing either a seed or pollen parent. So this information can be used for various different applications, and we can use this, we predominantly use this for quality control in breeding programs, where we can confirm the identity of propagated material and also look at relatedness between individuals, whether it be parent pairs or full sibs, half sibs, or whatever. Okay, so I'm going to just share a little bit about DNA fingerprinting, but just to orientate you beforehand, I am representing the genotype or the DNA fingerprint of a tree using these little blocks. All of those in the pink were inherited from the seed parent, and all of those in the blue were inherited from the pollen parent. And each row indicates a DNA marker. So you can see that at each marker, they inherit variation from the seed parent or from the pollen parent. So if we consider identity verification, if we take 4 trees, for example, and we generate DNA fingerprints for them, we can clearly see that those that 1, 3, and 4 have the same DNA fingerprint, but we can also pick up variation or differences in tree 2, and we can assign that to a different cultivar. So we can use relatively few markers to be able to pick up differences between individuals because they are supposed to either match 100% if they are the same cultivar, or they may have even 1 or 2 mismatches and then be classified as an alternative cultivar. So the fewer mismatches they have, the closer they are related, and the more mismatches we see between 2 individuals, the more distantly they are related. So, like I mentioned, we do not need that many markers to be able to distinguish between individuals when we are doing identity verification. So for this, we can use just maybe 10 markers or so is sufficient to be able to distinguish between individuals. When we consider relatedness, if we were to have a progeny sample with an expected seed parent and a pollen parent, here I'm representing the 2 variants at each DNA marker for the seed parent in 2 shades of pink, and the same with the pollen parent. If we're just considering 10 markers, we may not pick up differences between genetic variants coming from maybe a different pollen parent if we're just looking at 10 markers. But if we extend this, maybe double the markers, we may be able to identify some differences between these individuals. So in this case, we can see all of the pink blocks come from the seed parent, but all of the blue blocks, we have 2 here, they do not come from this expected pollen parent. So that means that this expected pollen parent is not the true pollen parent, and we can then go look in our reference set to see if we can identify an alternative. So the message here is, while we can do parentage analysis with a single panel of markers, it's not necessarily going to be sufficient resolution to be able to identify mismatches, especially when there's relatedness within the pollen parents. So in parentage analysis, where an individual inherits only 50% of the DNA from each parent, we need additional markers to identify mismatches. So up until now, we have been doing parentage analysis with a single panel of markers because that is all that we had available to us. But I'll tell you a bit about the development of our second panel now. Okay, so what have we done so far? I'm going to start at the beginning. So far, we have established a sampling and DNA extraction protocol for various different tissues. So to start with, leaf tissues, that's the easiest for us to extract from. And this is suitable for cultivar analysis, parentage analysis, and various other applications. We can also do parentage analysis from kernel samples, and also confirm the identity of rootstock if we sample bark samples from lower down in the tree. We can also extract DNA from pollen, and this could be used to identify the identity of the pollen donor. For example, if pollen has been collected from a specific tree and maybe it's been mislabeled and you want to find out what it is, we can fingerprint that and assign the cultivar identity to that pollen. I want to just mention that a DNA extraction from kernel samples and from bark samples is a bit more expensive, and that is because we use a different protocol to be able to grind up harder tissues, and this requires extra plasticware, extra instruments, and those things are actually quite expensive. So it looks similar to a coffee grinder, and it's a disposable little cup that we add liquid nitrogen to, and we pulverize the sample using that. So it does add quite a bit of expense to the DNA extraction for kernels and for bark. We've also established sampling kits. So many of you will have received these. We send out a pretty little box, It has a field sampling guide describing how you can sample and store and ship various different materials. And then whatever you need to sample, whether it be into a block or a tube or a Ziploc bag, the biopsy pen and silica to dry the samples as well. So when booking a project, you can just say you would like a sample kit, and we will custom pack that based on your project and send that to you. In terms of sampling methods, we have, as I mentioned, different types of tissues that we extract DNA from, and we have different sampling methods for each of these. So firstly, we have leaf samples, and this is the most commonly sampled material. For our DNA extraction, we send you a little biopsy pen, and we need 6 leaf discs cut to the size of that biopsy pen to perform our DNA extraction. So the best way to do this is to pick 6 leaves from the tree, and this should preferably be new growth, if at all possible, because the older the leaves, the more leathery they are, the longer they take to dry, and the harder they are to grind up. But we can still extract DNA from more mature leaves. It's just easier and quicker from new growth. So you can pick 6 leaves. You can stack those on top of each other and just use the biopsy punch to punch a hole through next to the midrib, not covering the midrib, and just transfer those into either a plate if you're sampling a lot of samples, or into a tube if you just have a few samples. And we will recommend the optimal sampling strategy depending on the number of samples and the application that you have. Once those are sampled, they are dried with silica. So if it's a plate, we give you a gas permeable seal, we give you a Ziploc bag and some silica, and you can pop the plate in there and allow it to dry at room temperature. If it's into a tube, we generally preload those with silica. You just pop the 6 leaf discs in there, close it, and ship it at room temperature. It's really important, if you have leaf samples dried with silica, to just ensure that if you are keeping them for a little bit longer, if you're not sending them to us directly, that the silica does not change color completely. So different silicas have different colors. The ones we use is normally an orange crystal, and when they are saturated, they turn a greenish kind of color. So if they are green, you need to replace them with additional silica, which means it means that they're saturated, they cannot pull any more moisture out of the leaves. So it's really important to avoid mold growing, to make sure that there's silica that can still be activated and absorb the moisture. And from there, they can just be shipped to us at room temperature. And once they have dried completely and become a little bit crunchy, we then use them in our DNA extraction. If we consider kernel samples, this is a bit easier to sample. Kernel samples can just be picked, popped into a Ziploc bag, and you just can add a bit of paper towel or tissue or something like that to absorb any moisture, and keep these cool at all times. So it's best to take a cooler box with ice bricks when you're sampling, and then to store them in the fridge until you ship them to us, and also ship them on ice in a cooler box, and preferably not on a Friday, preferably a little earlier in the week if possible. Bark samples are a little more tricky. So if we look at the layers of bark and wood, we are extracting DNA from that central layer between the inner bark, from the inner bark where it joins the sapwood. So we need bark samples that include that. So it's not just the outer part, it's the inner parts of the bark. And these can be sampled with I think it's called a hole saw, or with a bark punch or something like that. Or even just taking a carpet knife and cutting out a square, making sure that you get that inner bark as well. And these are a little bit more sensitive than the other samples. They should be added to a Ziploc bag again with paper towel and shipped to us as quickly as possible, because we want to try and avoid oxidation of that inner bark before we do our DNA extraction. So these must be kept cool at all times and shipped to us as quickly as possible on ice. Then pollen samples, we haven't done a lot of DNA extraction from pollen samples. This is my only cute little bug in my whole presentation. What we have tested so far is to take racemes, to dry them, and to shake out the pollen, collect that, and then do our DNA extraction from that. We have been requested in the past to see if we can extract DNA from pollen on the leg of a bee. I'm always happy to try anything. You never know what you can get. We haven't tried that yet. I do suspect that we may not get sufficient pollen to be able to do to get a good yield in our DNA extraction, but we're more than happy to try if anyone wants to send us a little bee. So it's important for us that pollen samples dry out completely, that there's no moisture in there. So we prefer to receive those, just pop them into a brown envelope, seal that, keep it at room temperature and ship it like that. These should not be allowed to sit with any moisture in them. So they can be shipped at room temperature and allowed to dry. Even if you have them on your desk for a bit, just keep it open, let them dry out completely, and then ship them when they dry. That's also fine. Okay, so that's just our sampling methods. We have quite extensive protocols in place. We have our sampling guide to give you an idea of what to do, a step-by-step idea. This can also be downloaded from our website, or you can contact us and just ask how to go about sampling. We're more than happy to help. Then in terms of DNA marker resources, I've already mentioned our one panel of markers that we are using already. This consists of 13 microsatellite markers. And for that, we are predominantly using it for identity verification, but we have also used it a bit for parentage analysis, but with the disclaimer that it's not entirely accurate yet. We are in the process of finalizing our second panel of markers. At the moment, it has 13 microsatellite markers, but that may change as we just do the last part. So this can be used for parentage analysis of seedlings, to reconstruct pedigrees, for various other applications. And something that I get asked quite often is the price difference between the two. So our DNA extraction, our single panel of markers, the price is made up of the DNA extraction, one panel of markers, and a 30% overhead, which covers general lab expenses. And then we have, for the second panel of markers, many people wonder if we need to do another DNA extraction, but we don't. We do one DNA extraction. We use our two panels of markers, and then we have the 30% overhead on that as well. Yeah, so it's not exactly double the price of a single sample, but it is more expensive. Then just in terms of where we are at with this panel, we have gone through multiple steps to identify our markers, to test them, to decide what fluorescent labels we want to add, and then to order fluorescently labeled markers. I'm not going to go into that, all the details behind that, but basically we have tested our fluorescently labeled markers, and we are just busy optimizing the input concentrations to make sure that we get optimal signal from the entire reaction. So this is a process that we like to comically refer to as panel beating. And that is what we are busy doing at this point. We can use the panel within another week or two, but we will continue this panel beating process over the next few months just to make sure that we get optimal signals. Okay, and then we're also busy working on the SAMAC cultivar reference set. So I've presented on this quite a bit before. Just to recap, a reliable reference fingerprint is really important for us to be able to establish or confirm cultivar identity. So if we have a specific cultivar, we prefer to sample 3 different trees, ideally from different sites. And if all of those match, we consider that to be a gold standard consensus reference DNA fingerprint for that cultivar. And this is what we're trying to build into the SAMAC cultivar reference set. So if we have a single sample from a single site, it's not as reliable. There could be labeling issues there. If we have multiple samples from a single site and they all match each other, that's a bit more reliable, but not 100%. If we have samples from 2 different sites that match each other, we consider that to be more reliable, but ideally we want at least 3 samples from 3 different sites, preferably from 3 different sources. So they shouldn't all come from a certain nursery and go to 3 different sites, preferably from different nurseries. And if everything matches each other, we are fairly confident in that reference fingerprint. So the reliability increases the more samples we have and the more sites that we sample. So just in terms of where we're at right now, our reference set consists of around 100 genotypes. Just over 40 are represented by single samples from a single site. And in these cases, we would like to sample additional samples just to be able to be a little bit more sure of the true reference fingerprint. And we have just under 40 that are represented by true consensus fingerprints, and we're relatively confident on those. And then we are also busy just identifying cultivars, or we have identified some cultivars where we are clearly seeing a bit of an issue, where there may be references from different sites or samples from different sites that do not match each other, and we see various cultivars or various genotypes all under the umbrella of a single cultivar, or we are seeing cultivars that are considered distinct cultivars, but some samples are matching from different sources. So those clearly need more intervention from us, and that's what we're busy doing at the moment, is we are going through, I've picked about 5 to begin with, to just go into a bit more detail to try and understand what is going on with those and sample further if need be. We've also focused a bit on increasing our human capacity, and any of you that have had field trips from our lab coming to your farm, you will have met a few of our team. So we have Darlene and Tolofela. They are both technicians in the platform, and we have directed them a little bit more toward macadamia and acacia, so that would be small growers and nurseries in South Africa. And they have also gone on a few field trips, and they are learning a bit more about the industry and will continue with this work going forward. And then I'm sure you all know Dr. Mary Ranketse. She recently graduated, and she is leading the molecular breeding aspect of our macadamia genomics. And she is also overseeing some activities, for example, in field trips and things like that, to try and integrate Darlene and Solofelo into the industry and teach them what she knows about macadamias as well. So we are building quite a bit of capacity around macadamias within the team, and we are all really excited about that. Then just looking at the application of DNA fingerprinting for macadamia, I've touched on quite a bit of this, and the video also touched on this. So in terms of identity verification, we can use this to ensure that the correct cultivars are planted, that you're receiving the correct cultivars from the nurseries, and even genotyping mother blocks that are used by the nurseries to make sure that they are cultivating the correct cultivars. This can also be used to verify the identity of rootstock. I'm not sure how valuable this would be to the industry, but we have had one project where this was requested. And then identity verification can be applied just to ensure that your orchard is laid out based on your preferred design to maximize the benefits of outcrossing. Because if you have individuals that you expect to be a different cultivar placed at strategic places to maximize outcrossing and they aren't the cultivar you think they are, you're not going to get the gain that you are hoping for. And then, of course, we can genotype unknown individuals where the cultivar identity is not known to see if we can assign them to an existing cultivar, or perhaps if they could be new cultivars that have not been fingerprinted yet in South Africa. Then with parentage analysis, the main application would be identifying seed and pollen parents. This could be just to identify the parents of a really good tree that's performing really well, or it could be things like ensuring that the correct pollen was, or the correct parents were included in a controlled pollination study. In forestry, we use this quite a bit to evaluate different controlled pollination techniques to see which ones are most successful. So I'm not sure how applicable that is in macadamia, but that is an option. We can also confirm outcrossing versus selfing in progeny. We can identify cryptic relatedness within parent pairs. So cryptic relatedness just means if you are not aware of relatedness between two individuals, but they actually are related. So that could be with cultivars where they come from a specific breeding program, and unbeknownst to everybody, two cultivars are actually siblings. So that can be quite interesting and important when you're planning outcrossing or cross-pollination. Oops, sorry. And then, of course, looking at family structures such as full sibs and half sibs. And this is really important if you're planning molecular breeding populations or something like that. Okay, so I've been talking for quite a while now. If you are interested in reading up about us at all, we have a website. I have to just mention that our website has been down for a little while. We've had some issues with just the licensing of that, where unfortunately someone unrelated to what we're doing made a decision to discontinue our website, and we are in the process of trying to get that resolved because that is how everyone connects with our platform. And then just in terms of the dates for the end of the year, our last day for sample submission, the last day we will receive samples, is the 6th of December. And this will just allow us to process everything before people go on leave for Christmas. And then we will open for sample submissions again on the 19th of January. So we try to time this with the sequencing facility on campus to ensure that we don't have any samples waiting if they have any issues with calibrating their instruments or servicing them or anything like that. So during this time, we do a bit of housekeeping as well. Okay, that's all I have to say. I don't know if there are any questions.
Thank you so much, Melissa. While we give the attendees a moment to type their questions in the Q&A, I think we can just quickly jump back to Prof. Gerda, if she's still there. Yes, I see she's there. A few questions that came in for you. So from Gerard, from a practical treatment and damage perspective, what is the difference between I assume it's the brown marmorated stink bug, which is not yet possibly an SA.
Okay, sorry. So what's the difference? I heard brown marmorated stink bug. I mean, we can't say anything. It's just that it has had a massive impact on various industries in the UK and in Europe. Whether or not it's going to really affect macadamias, time will tell. But I think there was another side of the question. Sorry, Lisa, I didn't hear you properly. But if you're asking about the bark stink bug, is that what?
It says marmot stink bug, so I assume it would be the brown marmorated one.
Yes. So, I mean, obviously, time will tell. We don't know. But then just the comparison that I showed, I can at least say the brown, so Cunomorpha nervosa, is a bark feeder, so it's not causing any damage on nuts.
Okay, thank you.
I just know, yeah.
Olivia?
I think, Gerda, the other part of that question was from a practical treatment and damage perspective, what is the difference? So I can maybe start and you can continue as you're the expert. I think control, probably I would assume similar things would work, but what we don't have damage thresholds. We don't know when and where they occur, what their life cycle is, but I'll hand over to Gerda to answer the question.
Okay, yeah. So sorry, I didn't then hear it properly. In terms of control, I think pretty much similar strategy than what we would do for two-spot will suffice. at least some of the good news is in terms of brown marmorated stink bug. If it enters, there's already commercial lures available. There's commercial attract and kill lures available. So that gives you that other option. But similar as what Rio said, Al-Shabaab has done quite a lot of work in understanding what is that sort of 2-spot threshold. And of course, it just being a different species, if we do understand that it's causing damage on max, that would be something that you would then have to sort of do for the brown marmorated stink bug. In terms of life cycle, I think the Green-Day model, if we look, I think 2-spot and Azara differ slightly. How much 2-spot differ from brown marmorated, I don't know. It would be very overlapping, but yes, for each and every new species, there are various things that you have to sort of revisit. But of course, I would say an insecticide is likely going to work on both. For a biological, for a EPF, I would think that you would need to test. But that is why it's good that there's so much research ongoing in Europe and in America. So there's a lot of published information that we can sort of grab and just test to see if it also works in SA. If it enters the country and if we do see that it is really attacking macadamias and if it becomes a problem.
Thank you. The next question is from Ernest. With regards to the sap beetle, what control protocols would you recommend in the orchard and drying bins?
So this is something we're going to hopefully start looking into next year. Orchards is very difficult. Remember that beetle sits in your nuts most of the time. So where we've done our winter sampling, we actually went in orchards after sort of harvest and cleanup, and they were sitting in the nut. So that's a big problem, because if you spray something, you're not going to be successful. If you want to spray, we need to understand life cycles. So you need to understand when is the beetle flying around. In terms of management, I don't know. It's something, just a conversation I had with Ria and Kayla the other day. There seems to be sort of specific heat treatments that can help. Our step number 1 is just confirming, so how many species, how frequent, is it really a problem? And then we'll sort of start taking the next step. So sorry, I can't really answer you, but a big thing to understand is the fact that if it sits mostly in the nut, the larvae, you only can sort of do a spray once you know the adults are flying, the beetles are flying.
Thank you. Then is it?
I see Ria wants to say something, I think.
I can maybe jump in there, but then I'm just going to point you in the direction of Kayla. I do think we have something registered for sap beetles, but as Gerda said, you would need to time it very specifically so that you actually get to them. But I think Kayla will maybe just send you an email.
Now from my side, I think we do have a registered product. I think it's carbaryl, but I'll just have to double check on that. And it might be for bark borers. I'll just double check and get back to you on that. Yeah.
Okay.
Yes, and please, bark borers are not sap beetles.
Yes, they're a bit different.
Next question is from Walter. What would you recommend for the control of lichens in macadamias?
So I don't see how lichens should be an issue. It is not something that needs to be controlled. It's a mutual sort of, it's a symbiosis between mostly a fungus and algae. So I don't see how you would want to control lichens. I've never heard that it could be of economic concern.
And then when do you guys close for the year and open in January?
Okay, yeah. So sorry, I saw when Melissa was sharing that information. The last day that we can take in samples will be on the 28th of November, just so that we make sure that we can actually process the sample. And then we sort of again open the second week in January. I think it could be like the 10th or the 9th. Let me just see if I can open up a calendar, or I can do it quickly on my phone, but it would be 10 or 11 Jan. 8, no, that's still December. It will be Monday, 12th Jan.
Perfect. Thank you so much. I
Thank
don't
you.
see any questions for Melissa, but in general, I think please everyone make use of these services. You can see there are many advantages to you. So really, really support it. I see there's a hand up.
Hello, good morning. My name is Aaron. I'm from Mozambique. So DNA and variants. How can DNA fingerprint help us confirm and monitor the purity or Macadamias variety CL in large variations?
I heard the first part of your question, but could you just repeat the last part, please?
How can DNA fingerprint help us confirm and monitor the purity or Macadamias varieties expel in large plantations?
Okay, I'm still not sure that I heard the last part, but you are asking how can DNA fingerprinting help you to monitor the purity of macadamia in your plantations? Is that right?
Yes, in
Yes.
large.
I'm sorry, it keeps on cutting out as you're saying the last part. But I'm going to try and answer you. So I'm not sure what cultivars are being planted there in Mozambique, but if it is many of the same cultivars, if we have the reference of that cultivar, we can genotype whatever you have and just verify that it is actually the correct genotype. If you're doing any propagation or anything like that, it would be important to genotype the tree that you're collecting material from in the first place, and then to genotype later on in the propagation process to ensure that you have the correct genotypes labeled correctly. There are many, many opportunities throughout the propagation process to make mistakes, and human error can cost a breeder a lot of money. So that is one thing. You can also verify what you receive from nurseries before you plant them out. And that is really important if you have a specific design that you want for your orchard, if you want to make sure that you have different cultivars placed amongst them to ensure that you have outcrossing. That is really important. If you're doing any breeding studies or anything like that, where you're actually doing controlled pollinations, that can be helpful to fingerprint that and just make sure that you have the correct parents. But perhaps you can contact me and we can chat a little further about that, because I would need to understand a bit more in terms of your setup and what you're growing there, and perhaps advise you a little bit more specifically.
Okay, thank you. Thank you.
Thank you so much. I think that's all time that we have now for questions. If you have any others, please feel free to type an email and send it to myself. Kayla or Aurea will either answer it for you or direct you in the right path. Thank you so much to Prof. Gerda Fourie and Melissa Reynolds. We really appreciate the time and the effort and all the hard work that you guys do for us and in these projects. It doesn't go unnoticed. So thank you.
Thank you.
Thank you. Bye, everyone.